Hi, I'm Dr. Joseph Gleeson, here to tell you about our new publication in Nature entitled Developmental Organization of Sensory and Sympathetic Ganglia. This was a publication led by Kang Harry Vong, pictured on the left, and Xiaoxu Yang in the middle, from the University of California, San Diego, Rady Children's Institute for Genomic Medicine, and the University of Utah. Our papers focused on neural crest cells. These are cells in all vertebrates that give rise to most of the peripheral nervous system. They're divided into three main types: the enteric nervous system, the sympathetic ganglia, and the dorsoroot ganglia. These cells are all born in the dorsal part of the developing neural tube, and they make a key step where they delaminate, which means they exit the neural tube and migrate into the presemitic mesoderm and populate the peripheral nervous system there, contributing to both. Here you can see the dorsoroot ganglia in red and the sympathetic ganglia in blue. And there's two main models about neural crest fate specification. The first is that these cells, when they delaminate, they're still retaining multipotency, and there are several publications that have documented this. And this is such that after a delaminating cell starts to migrate into the body, it's still capable of giving rise to multiple types of progenitor cells. The other model is more of a fate-restricted model, where upon delamination, the cells are committed to a particular fate, such as these cells going down the blue path and these cells going down the red path. And there are several publications to document this potential model, but obviously these are mutually exclusive models. So to address this, we took advantage of a new technology called somatic variant barcode analysis, where we identify individual mutations that occur in cells as a process of normal cell division and homeostasis. These mutations are generally benign. They don't cause any effect on the health or the survival of the cell, but they can then be used as a barcode, because all daughter cells deriving from the mother cell that has the initial mutation, like this yellow cell here, will contain that same mutation. So we can follow those mutations across the body in healthy individuals, healthy people actually. So this was a technology that we helped develop, and we can measure from many individual organs or cells or cell types across the human body the allelic fraction. That's the percent of each collection of cells that we sampled that contain that mutation, such as this green mutation. Here we have only 2 cells, and so we can calculate the allelic fraction of 0.05. Here we have a greater population, a greater percentage of the cells, and so the allelic fraction is higher.
I am Dr. Xiaoxu Yang from the Department of Human Genetics, University of Utah. So I will introduce how are we going to use mosaic variant barcode analysis to understand the distribution of neural crest-derived ganglia from human donors. 3 human body donors, we were able to recover hundreds of ganglia biopsies, sequence the whole genomes of many of them, together with samples from outgroup tissues. After machine learning-based mosaic variant detection from the whole genomes, we further quantified the allele fractions of candidate mosaic variants with massive parallel amplicon sequencing, and inferred the genetic similarity and lineage relationship of these neural crest-derived ganglia based on hundreds of mosaic variants from each donor. We observed that most of the stable terminal branches with high bootstrap values are consistent only between DRGs or SGs, which shows that most neural crest progenitors might have fate bias and gave rise to either DRG or SG populations exclusively. We also found that these ganglia from stable bootstrap branches always span across multiple spinal levels, which is a clear sign of rostrocaudal distributions of clones seeded by similar progenitors. Here is an example to elaborate on how distribution of mosaic variants teach us about cell fate elimination. As neural migration is a path of no return, we show examples here how the mosaic variants represent cell fate elimination. Note that as each cell acquires many different mutations, these observations may overlap. Cells containing those yellow mosaic variants acquired before delamination will be distributed to both left and right. Cells containing orange and green variants acquired after delamination are specific to samples from only one side. If delineated neural crest cells retain multipotency, as shown in scenario 1 here, bilateral variants should be observed in both DRGs and SGs with relatively equal representation. In a 2D dot plot, where X is the average allele fraction differences between the left and right, and Y is the average allele fraction differences between DRG and SG, they should show both X and Y coordinates close to 0. For unilateral variants, if they can become both DRGs and SGs, we would expect Y coordinates close to 0 and X coordinates not equal to 0. If delineated neural crest cells are phase specified, as shown in scenario 2 here, bilateral variants should be observed either only in DRGs or only in SGs, resulting in X coordinates close to 0 and Y coordinates not equals to 0. Similarly, unilateral variants should show neither X nor Y coordinates equals to 0. If we aggregate hundreds of informative variants from each individual, scenario 1 will result in an almost horizontal cloud of points, and scenario 2 will result in an almost vertical H-shaped cloud of. Our observation of hundreds of variants from 2 example human individuals shown here, you can see from the KDE plots and the distributions that this result basically supports scenario 2, demonstrating that delineated neural crest cells are likely phase specified at very early time points, forming a vertical distribution.
Hello everyone, I'm Vong, the first author of the study. I will tell you how we use different species to further understand the development of the neural crest. We wonder if the lineage and clonal relationship that we observed are evolutionarily conserved or specific to humans. So we used a similar approach to look into neural crest development in mice. In mice, we can use CRISPR-Cas9 to introduce artificial somatic mutations in specific genomic loci. Using 2 independent CRISPR-Cas9 barcoding mouse models, we found that the sensory and sympathetic cells mostly do not share the identical mutations, meaning that they are mostly derived from independent pool of progenitors. We also wonder if we are able to observe cell phase specification at a single cell level. So we performed single-cell RNA sequencing for the most neural crest cells. We found that a lot of the neural crest cells, they already express several genes that are known to drive either sensory or sympathetic fate before they delaminate and leave the neural tube. So these results suggested that cell phase bias for the neural crest progenitors emerged very early when they are still within the neural tube. We also performed an analysis that's called RNA velocity, which allows us to infer the lineage trajectories between different cell types. We are able to identify genes that show differential expression dynamics towards either sensory or sympathetic fate as early as embryonic day 8.5, which is a time point when the progenitors are still within the neural tube. Another observation that we could make from the human tissue study is that the developmentally related cells, they often come from ganglia of different levels. And one hypothesis is that the progenitors, they may migrate across different levels during development and spread the clones across different ganglia. So we tested this hypothesis by performing live imaging in a Quail embryo. We labeled neural crest progenitors with fluorescence, and then we follow live how the cells could migrate within the embryo for 6 hours. This is one of the representative videos. We can see that the cells migrate across different spinal levels. And by quantifying the migration distance, we observed that approximately 20% of all the progenitors could migrate a distance that is more than 2 spinal levels within the 6 hours. We also asked what kind of signaling molecules may regulate this process. By looking into the single-cell RNA sequencing dataset, we found that the receptors for a molecule that is called FGF are most enriched in the new Crest cells that are not yet delaminated. Therefore, we gave the embryo an inhibitor for FGF and observed the effect. In the control, the green cells, which are the new Crest progenitors, they migrate very normally, but in the embryo treated with FGF inhibitor, the cells no longer migrate across different spinal levels. Instead, they migrate out of the neural tube. So our findings suggest that FGF signaling molecules are required for keeping the progenitors within the neural tube and distributing them to the ganglia of multiple levels.
Thanks so much, Harry and Xiaoxu. In summary, using somatic mutations as barcodes, we found that the mutational clones are not shared between the sensory and sympathetic ganglia. This suggests that they are derived from mostly distinct progenitor pools. And our analysis suggests that the delaminated neural crest progenitors are predominantly fate specified, and that this fate bias emerges when they are still within the neural tube. And moreover, we found that these progenitors can migrate extensively along the rostrocaudal axis of the neural tube, distributing clones to different spinal levels, and that this migration depends upon FGF signaling in part. I would like to acknowledge all the amazing co-authors, in particular support from the Rady Children's Institute for Genomic Medicine with Stephen Kingsmore and Melanie White's group that contributed the work on the quail embryos, as well as many members of the Gleeson Laboratory and funding from the NIH, the Hillblom Foundation, the Simons Foundation, and the U.S. Korea grant. Thank you.